Hello, and welcome to the video lesson on cloning. Today, we're going to talk about two types of cloning. The first type of cloning is called reproductive cloning. Okay, and this is the cloning that you probably heard about in your life. Okay, the goal of reproductive cloning is to produce a complete organism, okay, that is genetically identical to the organism that you're cloning. So the example that was in the news or has been talked about is Dolly. So Dolly was a sheep, okay, that was a clone of another sheep, okay. We're going to talk about this process today and how reproductive cloning is performed. They've also cloned cats, dogs, mice. It is illegal to clone humans or even to try to clone humans for ethical reasons, which you'll see why in a second. Therapeutic cloning, okay, which is done not to produce an entire organism, but it's done to produce embryonic stem cells. Okay? These embryonic stem cells are said to be, if you remember from like the last unit, pluripotent, and they can develop into any other tissue type. Liver, heart, nerve cells. This is done with human cells. The goal of this type of cloning is that you'll be able to grow organs using your own cells. And we'll talk about why a little bit later, like in about 4 minutes, why it's better to have organs put in your body that are made of your own cells than opposed to cells that are made from someone else's embryonic stem cells or from an embryo that was made in a fertility clinic that is not being used, and you're going to use those cells, which are another person's cells, to grow a liver. Using your cells is better, and we'll talk about why. So this slide shows the process of cloning. Okay, the overall process is called somatic cell nuclear transfer. Okay, we're going to talk about why it's called that after we go through it. I suggest that you write all over the slide as I go through it. So this here is the sheep that we're trying to clone. Okay, the first thing you need to do is remove a body cell. Another word for body cell is somatic. Okay, from this sheep. Here they're taking it from the udder. So this here is the cell from this sheep. This is a female sheep. From her, you're going to take out an egg cell, and using a microscopic needle, they're going to pop out the nucleus, or denucleate the egg cell. So at this point, this is what we have. We have a cell that was removed from a sheep that we want to clone, and then we have an egg cell, which we pop the nucleus out of. We are going to bring these two cells together and fuse them using an electric shock. At this point, this cell is the same as a fertilized egg because it has a full nucleus, right? Which we'll learn later, that is diploid, or has two copies of each gene, and we're going to learn that in the next unit what that means. Okay? This, basically, what seems to the body, or seems to itself as a fertilized egg, is going to begin dividing. And if you remember, the first stage in embryonic development is cleavage, when the cells divide without growing. And then you will get an embryo. Okay? This embryo, which came from the division of the cells, or the cleavage of the cells, will then be inserted into the uterus of a mother, in this case, sheep. And then hopefully, this embryo will develop into a baby lamb. Okay? It did work. They named the baby lamb Dolly. So this clone here, which was produced by putting this embryo into the uterus of this mother, using the DNA from this sheep here, is genetically identical to this sheep here. Now, it turns out that it wasn't like they just did this once and it worked. They tried about 35, 40 times and got one successful clone. Hence why we don't do this in humans. Okay, we can't be making human embryos and only having one out of the 35 work. That's ethically questionable. Some people could say that doing this to sheep is ethically questionable. But anyway, let's go back to the name of this process of cloning, which is called somatic cell nuclear transfer. The reason why it's called somatic cell nuclear transfer is that you're taking a body cell from a sheep, that's the somatic part, and you're transferring the nucleus into a denucleated egg, and that's where the name comes from. Once this egg has the nucleus in it, it acts like a fertilized egg, divides and forms an embryo, and then you insert that embryo into a female uterus, in a female's uterus, and you hope you develop a lamb that would be genetically identical to this lamb here. Now, they've also done this process in other animals. One animal they did this in is a cat. And amazingly enough, or I love this name, they named the cat Cece, or Copycat. But if you notice here, this is the cat that they took the nucleus out of and put it into an egg of a female cat and into the uterus of, and then let that egg develop into an embryo, and then implant the embryo into the uterus of a female cat. And this is the clone. Notice that they don't look identical. And what this sort of revealed is that there are some things, such as fur color in cats, that are not under complete control of just the A, T, G's, and C's. These this is a calico cat, so it's got red here, white here, black here, happen randomly during development. When we get to Mendelian genetics, we're going to talk more about calico cats and why they can be both red, orange, and black, and how that coloring is not controlled by the A, T, G's, and C's. It's controlled by something called epigenetics, which is changes in gene expression not due to the A, T, G's, and C's, but due to other things. But the point is that sometimes when you produce a clone, it's not identical. It's genetically identical, but it may not appear identical because there are some things that are not under complete genetic control. Some of them happen during development or in environmental. Another thing that's also been shown with the clones is that the clones tend to have shorter lifespans. And the question is why? If we were doing this lesson in class, I would ask this question and I'd be like, if you knew this answer, I'd be amazed. But we're not in class. So the question is, why do animals that are cloned, or the cloned animal, tend to have shorter lifespans? And the reason goes back to something we learned in the last unit, is that when you're taking a cell from in this case a sheep, it's an old cell. It's already divided a couple times. And if you remember, what happens when cells divide, they need to replicate their DNA. And if they replicate their DNA, what happens is their telomeres shorten. So it's thought that cells from the clone, they start off with shorter telomeres than a regular embryo would. And then as a result, at some point in the animal's life, those telomeres get shorter quicker because they're already starting off shorter. And that leads to sort of death of cells, which can lead to death of organs, which then leads to the death of the organism. So once again, just to repeat, the reason why they think that clones tend to live shorter lives is that the cells that they get them from are old. They already have short telomeres. As a result, those telomeres start off shorter, and then eventually they get a point which causes the cell to kill itself. And if those cells die, they're part of an organ. That organ dies, and then if that organ dies, eventually, the organism will die. The last thing we gotta talk about in this lesson is therapeutic cloning. Now remember, the goal of therapeutic cloning is not to produce an entire organism, okay? It's to produce organs that can be used to help, in this case, we're looking at this example here, humans. So let's look at this process here. I suggest that you write all over it. So let's say, let's say this is a nucleus from my cell. And let's say for whatever reason, there's lots of reasons, I need a new liver. Okay? So what they would do is they would take a nucleus from one of my cells. Usually it's a skin cell, but it could be other cells as well. And then they would take an egg, probably one of my eggs. Okay? And they would insert the nucleus into this egg just like they did with Dolly, the sheep. Then, just like they did with Dolly the sheep, they would shock it, okay, and they would let it develop into an embryo. Okay, this here is a blastocyst, which we saw in the previous unit when we were learning genes and development. And these cells here are the inner cell mass. Okay, those are the cells that are going to develop into the baby. Okay, they are also said to be either totipotent or pluripotent, which means that they can develop into any cell type. Okay, that's why the cells of the inner cell mass are used, because they're totipotent or pluripotent. Okay, they can develop into any cell type. Now, at this point, though, if we were doing Dolly or we were trying to clone a human, we would put this embryo into a human's uterus. But we're not going to do that here. Instead, we're going to try to get these cells to develop into nerve cells, to liver cells, or pancreatic cells, and then insert them, like get them to go into those organs, and then replace the organ that is failing in the human. Okay? You know, we know that our body can do that naturally in development. So this is the point where we are now. How can we get these pluripotent stem cells to develop into nerve cells, liver cells, pancreatic cells? What genes do we need to turn on and off, and how can we turn them on and off? Now, the question is, why is it better, okay, to have your own cells, like this is the nucleus from my cell, and eventually if they got it to develop into a liver cells, they would put that cell back into me. Why is it better to do this with your own cells, as opposed to just using embryos? Again, like there are embryos in fertility clinics that are in this stage that are not used, that are just sitting in ice. Why is it better to use your cells and not just anyone's, embryo cells? And the reason why is your cells, if you grow organs using your cells, those cells, when they develop into the organ and are put back into your body, are less likely to be rejected by the immune system. Oftentimes when people get organ transplants, one of the dangers is that their immune system will attack it as foreign, and then it will basically kill the organ. When you get an organ transplant, since they don't use therapeutic cloning yet, usually you get on a waiting list. Like they don't really even use embryonic stem cells either. You get on a waiting list and wait for a living person to die that has a healthy liver or a healthy heart, and they quickly transport it to where you are. But when you get a transplant from someone's heart or someone's liver, they give you immunosuppressants, or drugs that suppress your immune system, with the hope that your immune system won't attack it and kill it, because it sees it as something foreign. But if you're using your own cells to grow organs, then your immune system won't attack it as foreign. Alright, that seems to be the end. Go answer the guided questions, and have a good day.